So microRNA are thought to be regulator of gene expression, and they can do that in a canonical manner, like binding the 3' untranslated region of mRNA, thereby regulating their function, or in a decoy manner. And for decoy manner, I mean it's the ability of microRNA to bind protein and prevent their function, and we reported that in Cell in 2010. So we found out that there are other microRNA which are capable of controlling multiple mRNAs at the same time, and that also by modulating either in a canonical or a decoy fashion. One of these is this MIR300, which works in progenitor leukemic cells and in stem cell in two different manners. In the progenitor cell, its expression is not required for the pathogenesis of leukemia. In fact, if you restore its expression like in normal cells, you impair leukemogenesis. In stem cell, instead, the levels of these microRNA are extremely high, like in normal cells, and we think that this microRNA is responsible for keeping the cells under quiescence stage, and therefore making the stem cell resistant to the current therapeutic drugs, tyrosine kinase inhibitor and so on.
Okay. Do you have any experimental data to support these ideas?
Yes, we do have experimental data. The experiments are both in vitro and in vivo, and there are also other experiments in animals that are currently ongoing in my lab.
Okay. Are there any results that you can share about these data?
Yes, there are. Okay. So what we know in the progenitor cells, when we overexpress, when we restore the levels of this MIR300, it blocks cell cycle, and then what it does, targets a pathway which is a pathway that inhibits a tumor suppressor. This tumor suppressor is a main regulator of normal cell function. It's called protein phosphatase 2A, and the pathway which I'm talking about is the JAK2 beta-catenin, SET beta-catenin pathway. This is a pathway responsible for the regulator of self-renewal survival of normal and leukemic cells.
Mm-hmm. And does this have then any direct impact on CML therapy now or in the near future?
I do not think that targeting one single molecule is the solution for the eradication of a disease. And I am really quite, skeptical about this single target molecule, unless, even BCR-ABL itself for CML, which was, we thought was the cause of CML, indeed at the end is not the only one. So there are drugs which can reactivate these tumor suppressors I'm talking about, PP2A. These drugs are called PAD, protein phosphatase 2A activating drugs, and they are capable of restoring the tumor suppressor activity and thereby totally killing the leukemic cells, but not the normal, in which the phosphatase activity of this tumor suppressor is extremely high.
Okay, well that's all the questions I have prepared. Is there anything that you would like to add?
No, I hope that to see more these drugs which activate the tumor suppressor in clinic soon.